production. You have the thymus, bone marrow, spleen, lymph nodes. Those are the major organs related to the immune system, but of course, all the organs, in some one way or another, via the lymph nodes, are involved in the immune system altogether. Welcome. This is just the first slide, so I didn't mention this much. On a smaller scale, of course, you have the cells of the immune system. There are still many cells being discovered and identified and characterized further in depth. The major classes of immune cells are T cells, B cells. Among the B cells, you have, of course, plasma cells. They're terminally differentiated B cells. You have dendritic cells, macrophages, neutrophils. And in the end, all the other cells in the body also communicate to these immune cells in one way or the other. So the immune system is much more than just a defense system. It is also a system that maintains the normal body function of other cells. And on an even smaller scale, you have the proteins. And there are many of those. Just to name a few, cytokines, complement factors, antibodies. Those are factors of the immune system. Intracellularly, you have, of course, many more proteins that are one way or the other involved in the immune system and in normal body homeostasis. I'm going to talk a little bit about the complement system today, one way or the other, and the major part of my presentation will be on antibodies. I will talk about one particular autoimmune disease. This is an overview of different ones, so that's what happens when the immune system starts to attack the body itself. Normally, of course, it's designed to defend the body against pathogens, bacteria, viruses, protozoa, all kinds of yeasts, of course, fungi. But it's supposed to leave alone the gut flora, for example, and it's also supposed to leave alone the body itself. And when that is not differentiated, self versus pathogen, you get an autoimmune disease. And this is one example where the T cells attack the joints. You can have skin diseases, you can have, as a result, various difficulties, kidney problems. Every part of the body might be affected by an autoimmune disease. For the doctors, that's a big problem because they have all a common mechanism, or a few common mechanisms, but the departments are structured according to the organs. And so in all of the hospital, there are some doctors that treat all kinds of different autoimmune diseases. And every one of them is rare. That's the problem. Altogether, they affect 5% of the population, so it's significant, 1 in 20. So in this group of people, unfortunately, we'll probably have in the lifetime a few of us that will have an autoimmune disease at one point or another. Fortunately, many of these are treatable, and there are huge progresses in the treatment of autoimmune disease. Many of them rely on the treatment that is also used for cancer therapies. The autoimmune disease might be caused by antibodies, also by T cells and all kinds of other mechanisms, but antibodies are a huge factor in autoimmunity. And for that, it's important to remember about the structure of antibodies. And these are Edelman and Porter, and these are the two guys that discovered the structure of antibodies. So these are the Watson and Crick equivalents of this protein. So they discovered how this was made up. And Rodney Porter found out that an antibody is made by 3 major domains, 2 Fab arms that bind the antigen, and an Fc, a crystallizable region. And the contribution of Edelman in the structure of the antibodies was that he discovered that each antibody is made of 2 heavy chains, here in blue, and 2 light chains, green here and yellow here. So they also discovered that here in the hinge region, that is generally 2 disulfide bridges that link the 2 heavy chains together, and there are also disulfide bridges that link the heavy and the light chain together. You don't need to remember the name, but these domains are of course crucial. So to recapitalize, these are the Fab arms binding to the antigen. It's for antigen binding fragment, and the Fc is for crystallizable fragment. Sometimes confused with constant domain. This is not the only constant domain in the antibody. The CH1 that is located here in the heavy chain, and the V and the CL, these are also constant. So this is also part of the constant domain. All of this is constant. This part is, in the top here is variable. So DFC is for crystallizing. So now for the functions of antibodies. Of course, they are crucially important for defending yourself against viruses, bacteria, fungi, protozoa, all the pathogens that we encounter. They're also very useful for passive immunization, that is, the injection of antibodies after an infection. For example, if you are a little bit older, your age probably, or onwards, and you have a wound, probably you will get either another tetanus shot, another vaccination to induce these antibodies, or you might even get, in some severe cases where there's already a clear infection, some antibodies against the tetanus toxin. There will be still produced in horses, I believe. So it's a passive vaccination based on the antibodies from horses. Of course, you need to understand the properties of antibodies to understand hypersensitivity reactions. And those are allergies and autoimmunity in general terms. So that's hypersensitivity reactions of the immune system are those where the antibodies, T cells, all the factors involved in the immune system are overactivated. They shouldn't respond, but they do. And that leads to inflammation, and in the worst case, an anaphylactic shock. For all of those, for understanding this, and this, you need to know about antibody properties, the specificity, what are the antigens that it binds to, the affinity, how strong does it bind, and particularly how fast does it bind to the antigen, and how long does it stay on the antigen. So there's two constants that are determined by the K on and the K off of an antibody. The stability is a huge factor. How long after it's produced does it stay in the circulation? Or how long after it's produced is it still present in the guts and in the mucosa if it's, for example, an IgA? And last but not least, the effector functions of an antibody. Mostly determined by the FC, so by the crystallizable part, and by the sugar residue that is located here between the two heavy chains, indicated in red. So antibodies are typically glycosylated, and even this glycosylation determines how active the antibody is and how strong the effector functions are of the antibody. So glycosylation is also a huge factor in immunity. I'm going to talk more about the effector functions in respect to the hypersensitivity reactions. And as you know, we have different antibody isotypes in our blood and in our body altogether. IgM is generally the first antibody that's produced after an infection. IgD is localized on cells, so that it's the B cell receptor that is involved in triggering the immune system and the regulation of the immune system. Then you have the various IgG antibodies. They are the antibodies that are in the circulation in the blood. So those are in concentration the highest in our body. The one that's produced more, so the highest production in the body, is the IgA. That's because the turnover of the IgA is really high. It's secreted to the mucosa and degraded really fast. We need IgA for keeping the homeostasis of our mucosa mostly, to keep the levels of different bacteria and bacterial toxin in our gut, for example, under control. And finally, IgE. That's an antibody isotype that is mostly efficient against pathogens like protozoa, parasites, worms, infections, and so forth. It's also, unfortunately, the antibody isotype that's hugely important in allergy. But any of these, in principle, can be related to autoimmunity. Generally, autoimmunity, the antibody isotype that we suspect first is an IgG1, because you see for the effector functions that it has, here everything that is in red is a strong effector function and pro-inflammatory, so it reduces the activation of the immune system. The IgG1 has most of those. On the other hand, the IgG4, for example, has very weak effector functions, so it doesn't activate the complement, it doesn't activate mast cells, and so forth. Question even is, why do we have such an antibody? because it has very, very little possibilities to do anything after binding. It does, of course, neutralize antigens, so it binds and might block them, but other than that, it doesn't do much. Also, we don't have much of it. We have only half a gram per liter in our blood, which is not much compared to the IgG1, which is 20 times more. As a reference, albumin is the highest concentration of protein in the blood. That is around 50 grams per liter. So that's still, antibodies are a huge part of your proteins in the blood. So the question is, why do we have such an antibody and what does it do? IgG1 antibodies are the cause of a disease called myasthenia gravis. And here's a very young patient, and it affects the muscles. So these are antibodies that damage the function of the muscle, and I'm going to explain to you how it does that. So this young girl has been asked to use her eye muscles, and that's simply done by asking her to look up and straight again a couple of times. And what you see after a while, after she'd been trying to do that, is unilaterally she gets the medical terms of ptosis, that's a drooping of the eyelids. So one of the eyelid's muscles stops working after a very short time. Of course, that's not very dramatic. Eyelid functioning unilateral is not a big problem, but this disease might spread to the rest of the body. Medical doctors speak about generalization of the disease, and of course you need your muscles for a lot of important functions of your body. The most important function to stay alive is breathing, and when that is affected, the patients do need to be hospitalized and ventilated mechanically for a while until the other treatments start working. So it's a potentially life-threatening disease. Actually, the most life-threatening in the old days, so this is in the old days before treatment, this was lethal in a large proportion of patients. Why was that? Simply because people couldn't cough properly. When you can't cough, you use your muscles. When you can't cough, the mucosa, the bacteria stay in your throat and in your bronchia, and you get lung infections after that. So the myasthenia gravis patients, before treatment was available, died very often of pneumonia. So it's a potentially lethal in that way too. How is the disease caused? It's a problem of the muscle, as I said, and in particular of the neuromuscular junction. It's a connection between the nerve and the muscle. And here, this small piece here, this is the neuromuscular junction. Each muscle fiber, each muscle cell has one neuromuscular junction. And this is what tells or communicates to the muscle that it should contract. This is what they look like. You have here in green the nerves, and in red you have stained the neuromuscular junction with the toxin, Bangra toxin. Why do we use that? Very simple. There's a snake, a Bangra snake, a very small type of snake, but there's a very potent toxin. And when it bites you, this toxin binds to your muscle, and in particular to the neuromuscular junction, and blocks acetylcholine receptor. And the effect of that is that it will paralyze your muscles. And you need very little toxin to paralyze a whole a muscle and even a whole person very quickly. So this is a very lethal, very small snake. When we label it with a fluorescent dye, and we use a safe concentration of the toxin to handle, of course, we can very easily find the neuromuscular junction in the muscle. In fact, you don't even see any background of the muscle fibers here, but you can expect every single neuromuscular junction being located in a separate muscle fiber. They run horizontally in this section. This is in larger magnification. Here I also stained the subsynaptic nuclei, or all nuclei, in fact, with a dye. In this case, it's Hoechst. You can also use DAPI. You find that when you look at it at the expression level of the different subsynaptic nuclei, that they specifically produce the proteins that are needed for this structure. So it's quite interesting. There's within the muscle fiber, there's a differentiation of the different nuclei. Muscle is special in that way. There's a cell which has many nuclei. It's not very typical, but that's because a muscle fiber is composed of different satellite cells that are fused together. In the end, they share the same plasma lemma, the same plasma membrane, but they have many nuclei. But these nuclei are specialized. So in fact, they belong to, I could say, a different cell type. When we stain also for the nerve, you get here a third color. The blue staining is for the vesicle acetylcholine transporter. And here, this is a nerve, and adjacent to that is the acetylcholine receptor in the muscle fiber. I'm going to explain to you how this all works. There's a third protein that I would like to introduce. That's the receptor-associated protein of the synapse called rhabsin, and that colocalizes exactly with the acetylcholine receptor. And you see the yellow staining here. That's composed of the red and the green here. And just next to that is a nerve. In the disease that I just introduced to you in myasthenia gravis, and this is an animal model of it in a rat, you only find the nerve proteins left, and all the postsynaptic proteins have gone, or to a large extent have gone. There's a very little left, so you only found the nerve intact, and the postsynaptic structure in particular is gone. So how do antibodies lead to such a destruction of these receptors? And this is one mechanism, generally very important in defense of against bacteria. So antibodies do bind to surface receptor. Normally this would be a bacterium. In this case it's unfortunately the muscle. And there's a protein that binds to it, it's called C1q. And this activates a rather complicated cascades of different molecules that bind to each other. After C1q binds C4, then C3, and then the numbering is consecutive, so it forms C5. And this is the first protein that integrates in the membrane, then together with C6, C7, C8, and C9. And C9 is polymerizes and forms this complex, which is a pore. And this is quite a big pore, actually a hole in the membrane, and it allows basically all proteins of the cell through. So even a complete antibody potentially enters the cell, and all the cell components can go out. The biggest problem, however, is that the cell, at least in this region where this happens, loses the membrane gradient of the electrolytes, and therefore loses the membrane potential. So the potassium that is normally located inside of the cell goes out, and the calcium and the sodium goes in. So this changes the membrane potential. Calcium going inside of the cell is, of course, a second messenger. And what happens after that is that this leads to the genes that are related to apoptosis. So this whole structure below will get dysregulated. The subsynaptic nuclei will start doing all kinds of things that they first try to cope with the damage, and in the second instance they might stop functioning altogether. So this is the complement activation. So this is what it does. Normal function is, of course, is not the induction of apoptosis. Normal function is to punch holes in bacteria. And how the bacteria cope with that is the second question. So this is what it looks like on red blood cells. You can do that taking them from sheep. And what you see here, those holes here, the black dots, correspond to the membrane attack complex. That's this green complex. And you can see that it really, like I drew it here, sticks out of the membrane by looking at it from the side. You see that it's really this pore that forms there. So it has, compared to the red blood cell, it really has a significant size. So you can really imagine that all kind of molecules go in and out, and that's bad news for a cell. I have to say, it's not that the muscle cell dies altogether. It's just that the subsynaptic region of the muscle cell, which you see here, depicted, gets damaged. So how does this structure function normally? You see here the neuromuscular junction. You have here acetylcholine receptor located inside of vesicles. This is what a normal synapse looks like, also in the brain. Postsynaptically, you have a high density of neurotransmitter receptors. They cluster together. And what happens when there's an action potential in the nerve terminal is that these vesicles start to fuse with the presynaptic membrane and release the neurotransmitter, in this case acetylcholine. In the brain, it might be dopamine or serotonin or all the other neurotransmitters that are there. They bind to the neurotransmitter receptor, to the acetylcholine receptor, and that will normally lead to the contraction of the muscle. I'm not going to explain to you how that works because this is more related to immunology. Anyway, the membrane attack complex forms here, and that leads to the damage that I explained to you before. There's a second mechanism that is important, and that's the internalization of membrane receptors caused by binding of antibodies. So how do antibodies do that? This is the process called antigenic modulation. They bind to the receptors, and then they lead to an endocytosis of the part of the membrane together with the receptor. This is then internalized and degraded in the lysosome. The result of that is that there's less membrane available, maybe that the cell could cope with because the muscle fiber is so long, but the number of receptors then is reduced. And that is a problem. So knowing this, how could you design a specific therapy to prevent these damaging mechanisms? And there's going to be two strategies that I'm going to explain to you that we tried in the lab. The first one is to design antibodies that bind and block sites that are normally accessible to the antibodies, and therefore do not cause acetylcholine receptor modulation. So these are the competitor antibodies that are mentioned in the title. A second strategy that we've tried and successfully, unfortunately is not usable in humans, but it at least works in animal models, is to anchor the acetylcholine receptors in the membrane by a protein that is naturally already there, which is called rhabsin. So the overexpression of this protein will fortunately lead to anchoring of the receptors and stabilize the receptors against internalization, despite the fact that there's an antibody that tries to modulate these receptors. So how would it work? Normally you have the acetylcholine receptors in the membrane. We use different, more simple scheme here. There's some rhabsin available, but not enough to anchor them all together. And the idea is if you add additional rhabsin to it, you can complete this matrix and then prevent that the antibodies after binding have the chance to modulate the whole complex, because it's simply too big and too much fortified against the modulation. So how would you do something like this? Here you see the muscle fibers running diagonally, stained for rhabsin in green, and the acetylcholine receptor in red. The colocalization, of course, results in the yellow color. You see that this fiber is overexpresses rhabsin and has increased amount of rhabsin localized in this fiber. How we do this? We inject a plasmid encoding for the rhabsin in the muscle fiber, and then we electroporate. So we add quite a huge current over the muscle externally by electrodes. Use 200 volts per centimeter, typically because it's a little bit less than a centimeter thick in the muscle. We have to apply 150 volts for a very short time, obviously, 20 milliseconds per pulse. And this punches holes in the membranes of the muscle fibers and allows the DNA to enter. If it enters, it is the genes on the plasmid are transcribed in a normal way. In this case, it's a viral DNA with a CMV promoter. It works very strongly in any type of cell. It would also work in ours and in basically all cells, and therefore would lead to the expression of any protein that is encoded in the cDNA, in this case rhabsin, and we find it then after a very short time. You see that most of the muscle fiber has been transfected because we used to report the plasmid, in this case beta-gal, and you see that more than half of the fibers have been transfected, more or less. Of course, this applies only to one single muscle of the animal that we're using. We couldn't possibly, with this technique, transfect the whole body. For that, we would need viruses that enter the muscles. That's also possible, but that's a type of gene therapy that is not entirely safe yet. So that is something maybe for the future. There are huge developments in the viruses that can be used for gene therapy. So it might be possible to apply this in the future. But does it help? That's the question. This is the control muscle, where you see some rhabsin and some acetylcholine receptor in all the synapses. When you add rhabsin, you find a little bit more, maybe 20% more. When we induce the disease in the animal with antibodies, just by simple injection of the pathogenic antibodies, you hardly find any acetylcholine receptors in the muscle left. When we add the rhabsine, we do. So it has been protective because this is from an ill animal. This is one muscle that was normally affected, and this was the only muscle in the body that we had been able to protect with the rhabsine, and that works. So the mechanism works. Do we also prevent the complement activation? The answer is no. This is an ill animal. You find no receptors, but you do find the membrane attack complex that punches the holes in the muscle fibers. That is still there. In the normal ill animal, that's to be expected. The same is true also in the ill animals. A huge amount of membrane attack complex, basically as we expected, but in this case also the acetylcholine receptors are still there. So in fact we have stabilized the acetylcholine receptors, but we have not prevented the membrane attack complex. So the pores that form in the membranes are still there. So does that then, in combination, we have the receptors, but we do have membrane attack complex, lead to disease in this muscle? And the way to analyze that is by electrophysiology. And this is a standard test that is also used in the clinic. It's a decrement test. What is done is a nerve is directly stimulated electrically, and you measure the compound muscle action potential. That's something that is you might not be aware of, but it's a normal phenomenon. If you contract your muscles, each of the muscle is briefly depolarized, and you can measure that very simply by putting a surface electrode on your skin above the muscle. You can also use a needle to punch into the muscle, but it's not even necessary. Everybody produces a small amount of electricity by simply moving. It's for those of you that like science fiction, the Matrix, the idea that is for generating electricity, that's that. The movie is unrealistic because the voltages that are produced are tiny. It's ridiculous in that way. But it's very well measurable, and when we do activate the nerves, we do get also depolarization of the muscle, and normally every time that we do that, the depolarization should be the same. Because each time we want to move, we will move in the proper way. That's different for patients with myasthenia gravis. They will be able to do it once or twice, but the third and fourth time this becomes already significantly less. And the reason for that is that also the nerve terminal gets a little bit tired. Normally we don't notice anything of that. So what happens is that the nerve terminal releases a little bit less acetylcholine after the third or fourth depolarization, and after that it stabilizes. And normally the muscle has a safety factor, that is, it has 5 times more acetylcholine receptors than is needed for making normal action potential. And some of the fibers don't have enough, and that's why you see the direct effect of the decreased amount of acetylcholine. So this is called a decrement, this drop. And this is something that we found only in the diseased muscle, and this is the effect that we found in the treated muscle. So we were able to prevent the decrement by adding the rhabsine. A completely healthy animal would look exactly the same. To understand that these peaks are put next to each other, in reality they come one here, one here, one here, so the time in between those pulses would be much longer. So they're just stacked on top of each other for visualization. So what could we do to also prevent the complement activation in the muscle? And this is something that is getting quite a bit closer to clinical relevance. I said the gene therapy is probably still science fiction at the moment. In the future might become possible, hopefully. But this is something that is actually very realistic, and we are thinking now how to proceed with the results that I'm going to introduce to you now. So I mentioned that the IgG1 antibodies are the antibodies that cause the disease in these patients by complement activation and by modulation. An IgG4 antibody, as opposed to that, they are different in their Fc part, and for that reason they don't activate the complement system. That was the start. So the idea was we could probably use the IgG4 antibodies for protecting the neuromuscular synapse by adding IgG4 anti-acetylcholine receptor antibodies in the circulation. They would subsequently bind to the receptors and in that way prevent that pathogenic antibodies bind to the same epitope. This is possible because these antibodies have a very specific site that they would like to bind to, which is called the main immunogenic region. It's rather specific in the patient, not 100%, but if the G4 antibodies bind, they would also protect a large area of the receptors that the pathogenic antibody subsequently could not bind to. And that was the idea. In our experiment, we used 2 monoclonal antibodies with exactly the same specificity. How would this work then? The main principle would be, and this is actually true, that the C1q molecule cannot bind to the G4 antibodies. The G1 antibodies don't bind, and therefore you would prevent disease. We had monoclonal antibodies from patients, and they're very specific for human muscle, at least for primate muscle. So the only animal model that we had available to test this hypothesis was a primate model. And this is something that you should not easily consider. It is a very unpractical way of doing research, but sometimes it's the only possibility. And in this case, the disease that we induce in the animals is only for a few days. So it's ethically acceptable, at least in my view, to do this. So what we did is we injected IgG1 antibodies, which activate complement and are therefore pathogenic, and the G4 antibody, which should be protective. Before you do any such experiments, you want to test in vitro the properties of the antibodies. Do they bind? And yes, they bind in exactly the same way, with an affinity. The affinity is normally measured as a half maximal binding of an antibody around, you can see here, 2 nanomolar. Small sideline on this. This experiment was not properly done because you have to use always, you see that I used 2 nanomolars of acetylcholine receptors. In this way, the lowest affinity that I could possibly measure is around 1 or 2 nanomolar. You should, if you suspect that the affinity is even higher, use much lower concentration of the antigen, and then you can measure lower affinities. So this is maybe an underestimation of the affinity. So for those of you that are interested in antibody kinetics and mathematical models, maybe you can come to me and I'll explain. But for all of you, as a bottom line to remember, if you want to measure the strength of the antibody binding, you should use as low as possible concentration of the antigen as possible. And this is another way, because I wanted to measure the competition. I labeled one of the antibodies with a flag tag genetically, so by putting at the C terminus the amino acids that correspond to the flag tag, and this is also produced recombinantly, genetically modified the plasmid. So this antibody could compete in this same concentration with the IgG1 as the IgG4. So when I had an excess of these 2 antibodies, the binding of the antigen to the flag tag goes down in exactly the same way. So this is a competition assay. Next thing that you want to check before you proceed is, do these antibodies activate the complement system in the way that you expect them to? And for that we used the 2 pathogenic, or the 2 antibodies that bind to the autoantigen and 2 controls. You can see that the IgG1 antibodies bind to C1q. Does it also activate the complement system? That's answered here. The next step is C4, and you see that only the G1 antibodies, but not the G4 antibodies, bind to C4. And the crucial step in complement activation, right in the middle of the cascade, is C3, and there you find exactly the same thing, at least in vitro. One thing that I just would like to remind you again of is that there's one pathogenic property that we didn't abolish by making these G4 antibodies, and that's this. Because our antibodies were still bivalent and could modulate in exactly this way. So was this the case? And I measured it. Yes, unfortunately, both antibodies, as I made them in vitro, modulated the receptor in the same way. The control antibody, IVIG, that's polyclonal human immunoglobulin G. It's used sometimes also for therapy of autoimmune diseases, didn't do anything. But these two antibodies at a very low concentration degraded acetylcholine receptor in vitro in the cell line. So I have rhabdomyosarcoma, which is a muscle tumor cell expressing fetal acetylcholine receptors. And in that way I can test the antigenic modulation. And you see that both antibodies in principle have this pathogenic property. So for testing the immunotherapy in the rhesus monkeys, I used three groups. A pathogenic antibody here, the G4, this is the medication alone. And of course the most crucial group is the two antibodies together. This is the decrement that we get in the animals after adding the G1 antibody. The G4 treated animals, this is a nice horizontal line, no decrement. These animals were not ill, and in the combination we also didn't see any signs of symptoms. The conclusion, this is probably not 100% true, at least we cannot conclude it from these data, is that there's acetylcholine receptor loss here. Well, this is certain. No acetylcholine receptor loss, that might not be entirely true, because we still have the safety factor, so if there's a 50% loss, we probably wouldn't have noticed it with this method, because it's insensitive. As a matter of fact, you might lose up to 80% of your acetylcholine receptors before you notice anything. And then when you lose 85, you get completely ill. So that's the dilemma that we have in this clinical test. For that reason, we also did immunofluorescence analysis in the same way as I showed it to you before. And you see that the G1 treated animals have a completely destructed neuromuscular junction, and in the combination, the structure is nicely intact. Another way of looking at it is by ultrastructure, a little bit complicated to interpret, but what you can see here is a nerve terminal, and this here is for example, a small vesicle with acetylcholine. Here's another one. These are mitochondria. This is the presynaptic membrane. This here is a synaptic button. And the postsynaptic membrane are these cells, but it's completely degraded, so that's why it's so difficult to interpret. Gets a little bit easier here. Here again you see the vesicles. This is the mitochondrium, it's not so nicely fixated. This is the presynaptic membrane running around here. And this is the postsynaptic membrane. And here the dense part of the membrane, this is where the acetylcholine receptors are located. It's so thick and so dark because the proteins occupy a lot of space in this staining. Whereas normal membranes like this, just the lipids, are much thinner. So you can really tell that the acetylcholine receptors are still intact in this condition, despite the fact that they probably were modulated. And this is a combination, the same picture. The nerve terminal is not so nicely fixated, but you can see here the postsynaptic membrane with all the acetylcholine receptor is nicely intact. By the way, these are the sarcomeres, these are the contractile elements of the muscle fiber. So this is really just a small, very small piece of the muscle. If this was a complete muscle fiber, it would extend all the way up to the back in the other side. Probably, I should say, well, I have to calculate. I would estimate more than 50 meters or something. I did another experiment, and this is the antigenic modulation experiment with the rhabdomyosarcoma cell line to test how strongly the sera was still modulating the acetylcholine receptor. So the purified antibodies modulated quite strongly, as I said. This is the pathogenic properties, even of the G4 antibody as it was produced. Normal monkey serum doesn't do anything. Animals treated with the G1 antibody have less acetylcholine receptors than these, obviously. There's this property that you see back here in the serum. G4 treated animals, that was very surprising. In the sense that the antibody that we injected, this one, modulated very strongly, and after injection it lost that property completely. So we thought about several explanations, like wrong dosing or the antibody is unstable. But then we did a control experiment. We combined this serum with an extra bit amount of this antibody, and then we found very little acetylcholine receptor left. That's normal, to be expected. But here we really showed that this serum had some protective properties, because when we combined this antibody amount which is pathogenic together with this serum, we also didn't find any degradation of the receptors. And this was very unexpected and could only be explained by the mechanism that had been found by a group in Amsterdam, a group of Kob Aberse and Janice Schumer. They found that G4 antibodies have a very specific property that is not shared with other human isotypes, but in other animals, and a few other animals also exist, in rabbits for example. Antibodies can fall apart into half molecules that are composed of a Fab arm and half of the Fc. And another G4 antibody can obviously do the same. And then they can recombine, making these what I could refer to as chimeric antibodies. It's not, maybe better I should call them hybrid antibodies. Hybrid G4 antibodies that are composed of two different Fab arms. And that's unique for human antibodies, because they're supposed to all look like this. At least before we published the paper, everybody believed that they should look like this. In fact, that's not true for all antibodies. For G4, it's not. They might look like this. In fact, nearly all of our antibodies in the blood that we have are chimeric antibodies, hybrid antibodies. They're not stable, of course. They might fall apart again and recombine with a third antibody and a fourth, and so on. So each of these conformations only exists transiently, and the turnover is once an hour or so for each antibody, so very fast. For that reason, any binding partner is a transient binding partner, and it very rarely will be a specific second antibody that it will pair with. And the antibodies then will be functionally monovalent for that reason, because only one arm is specific for something on a cell. So in contrast to the antibodies that are injected, the antibodies that are in the circulation after a short while are monovalent. And that's relevant for the antigenic modulation, because the original antibody was able to bind to antigens in the membrane, whereas the resulting antibody is unable to do this. Therefore, as I said, functionally monovalent, is not able to modulate. Not sure if this is working. It might. No, it's not. It was a short movie saying this. Anyway, so I have to change the scheme of what's going on after injecting the antibodies. G4 antibodies bind in a monovalent way to the acetylcholine receptors. They do not modulate. I think I put this. So therefore, they don't internalize the receptors. But they still prevent, of course, the binding of pathogenic antibodies to the same epitope. So we published this as a big joint effort. It was published in 2007. And it is relevant because there are already a couple of G4 antibodies that are used clinically. There's one that's used for the treatment of MS, multiple sclerosis. And that's relevant because after injection, it will pair with the antibodies that the patients already have. And in each patient, this might lead to a different result. So it's somewhat less predictable than other antibody isotypes. On the other hand, it still has favorable properties, like it doesn't activate the immune system as strongly. And the main conclusion that we got from our results is that the antibody is anti-inflammatory because it is able to compete with pro-inflammatory antibodies. So this brings me to the kind of conclusion of this presentation. And that is that complement activation works together with antigenic modulation in myasthenia gravis and probably in other antibody-mediated diseases. If you have complement activation and you have antigenic modulation, both of it together, you will get disease. If you prevent either one of these two, for example, you prevent the antigenic modulation by overexpressing rapsin, but you still have complement activation, you will also not have disease. You can also prevent the disease by complement inhibitors, by using proteins that inactivate the complement system. With the G4 antibodies, we did both. We prevented the modulation because our antibodies were monovalent, and also we prevented the complement activation because the G4 antibodies don't do that. So that doesn't completely allow any conclusion about that. But the complement deficiency and the complement inhibitors very strongly suggest that complement is also absolutely important for getting the disease. The next step is making these type of antibodies. I've already done it. Also, it seems to work. Hopefully, we'll publish that soon. The issue with these antibodies is we can't inject them like that because we have to. These antibodies have to undergo a Fab-arm exchange in vivo, so they're pathogenic for a short while. We don't want that. So the next step is to inject an antibody form that is not bivalent when we inject it. And one possibility is to deleting the hinge, that is this region. In that way, it doesn't pair with another half molecule and stays in this conformation without pairing with the second Fab-arm. This is an antibody format made by Genmab, and for those of you that are interested, you can maybe Google Unibody, which is this antibody format, and you will find some interesting information. I can already say one thing about it, that seems to be very stable, so this is an interesting thing to use. For me, it is very interesting because it is naturally monovalent, and it will still bind obviously with a long half-life. So we this basically reproduce the same type of experiments that I showed to you earlier, positive results, and hopefully this is something that might go to the clinic at some point or another in order to prevent acute or to reduce acute disease. In those cases of patients that have to go to the clinic with respiratory problems, for example. Should I explain this one?
No.
Yes, I can. I think this should be on my last slide. Oh, I had more results about it. Yeah, I'll explain. Do we have time?
No.
Okay. This is an experiment that I did with a little complicated plot. Going to walk you through. I combined different amounts of pathogenic antibody with different amounts of the unibody, that's the monovalent antibody form. And the question was, can we prevent the modulation with this unibody as well? So when we increase the dose of the pathogenic antibody, and if you look at the amount of acetylcholine receptor left on the cells, you will see that it, with increasing amount, will decrease. Well, that's to be expected. Then when we combine that with different amount of unibody, for example, with 0.3 nanomolar, then you have to go to this point, then we are able to recover this amount of acetylcholine receptors. So for each combination, you get one single plot, and you can always follow, if you start with this amount of pathogenic antibody, how much unibody you will need to get back to the normal levels. And if you ever have two drugs that interact with each other, one pathogenic and one that is recovering the effect, and it's a dose response where you can plot the product of the two concentrations, and you will get a plot like this. Not commonly used, but I think you can see nicely the effect of the pathogenic antibody, and here the effect that you get when you add the unibody to that. Here, you'll put the PowerPoint on the layer board. Wilfried will, so you can have a look at that later. How did I test this in vivo? We injected antibodies on 3 days, day 0, 1, and 2. Took blood samples on different days. We took a biopsy before and after. And we did another more sensitive test, which is called single fiber electromyography. And we did that pre-treatment, so before treatment, and after injection of the antibodies. And the injection of the antibodies included both the unibody and the pathogenic antibody, G1. So how did we measure now in a more sensitive way? I told you the decrement test is very insensitive. This is very sensitive, but also more difficult. When you stimulate the nerve inside of the muscle, it branches into a lot of parts. And if you do that very closely to the muscle fiber, but not exactly on the muscle fiber, you can then subsequently record the membrane action potential of the muscle fiber with this very small needle. If you have a sufficiently small surface electrode of the needle, you will only record very few action potentials of individual fibers, single fibers. And if you do that repeatedly, then the action potential should always reach at the same time. Well, if you stimulate at this time, it will take some time for the nerve and subsequently the muscle to conduct the action potential. But eventually in a normal muscle, the action potential occurs always at the same time. This is an overlap of different stimulations. So this is showing the same. There's a slight variation, and the slight variation is because a normal nerve terminal releases different amounts of acetylcholine. Sometimes a little bit higher, sometimes a bit lower. This is a normally distributed amount. But this small variation will lead to a very small variation in the time which it takes to make the action potential. You see that they're not all identical. If you have different levels of stimulation, for example, or different levels of acetylcholine that's released, you get very, very small variation of the time which it takes to measure the action potential. When you have less acetylcholine receptors, you're very close to what is called the threshold, the triggering threshold of the muscle fiber. And then it might occur that you have just enough strength of, or just enough acetylcholine to make an action potential, but it will take much longer because you're just trying to get, try to imagine, you run with your bicycle, you try to go up the hill. If you're not fast enough, you could go over the hill very, very, very slowly and then over it eventually. If that happens, the action potential might reach much later. Yeah, so you've just gone over the hill with the bicycle very, very slowly. Eventually you do get down the other hill and you arrive at the other side where you measure, but then much later than your fellows that have just gone over the top of the hill a little bit faster. When you get this, you get what's called a jitter. The action potentials don't arrive always at the same time, and this happens especially if you have less acetylcholine receptors. And that's what you can measure. And this is a much more sensitive way of measuring action potentials in the muscle. So normally, the jitter of a normal muscle fiber in each of us, hopefully, is between 10 and 20 microseconds. So the variation which it takes each time that you try to contract a muscle, it's sometimes 20 microseconds faster, sometimes 10 microseconds slower. This is a typical variation. Wouldn't be much, right? If I contract my muscle and it takes 10 microseconds longer, it's not a big problem. Anyway, this is the average of many muscle fibers. In the unibody group, this did a little bit something which we quite understand, but it was very small. We didn't have many animals, only 2 in this group. When we treat the animals with G1 antibodies, we do get a significant increase of the jitter. That's to be expected, that's the pathogenic effect. So sometimes it takes up to 80 microseconds longer to generate an action potential, which is already significant. When we then add the unibody to that, we don't get back to normal, but we do significantly decrease the jitter. So that's a significant improvement, and that was very promising for us. What you can also do, and I didn't show that, you can measure the blockings. Sometimes the action potential doesn't reach at all. You do stimulate, but the muscle fiber simply doesn't have enough acetylcholine receptors to work with and doesn't make an action potential. Then you get a blocking, and this graph looks exactly the same. I did something that's not normally done, but you can also measure the decrement, even though it's only considered significant when it's over 10% in an individual. You can measure it in different animals because you do get a number, even if it's below 10. Normally the decrement is around 0 in control animals It's slightly higher in ill animals, or in antibody-treated animals, pardon. Here in the G1 treated animals, it's significantly higher. So basically, that recapitulates the result of the graph before. And this is, you can measure two parameters, the amplitude and the area, that just basically shows the same. So, I think that is really the end now. The receptor cross-linking by antibodies leads to internalization. That's not typically known, but it's important. Also, for example, in cancer therapy. And G4 antibodies do not modulate membrane receptors. I think this is a take-home message that you can read.
Ik wou nog zoveel opschrijven over zijn laatste.
So, why is this important? When you do design an immunotherapy for another disease, you should remember that there are different antibody isotypes that you choose from. In cancer therapy, very generally, it's G1 antibodies. They have a long half-life, but this is not the only application. If you think about autoimmunity, if you think about neurodegeneration, there are all the other antibodies, isotypes, the human ones that you can choose from, and they're becoming more useful. And this is something of the last years, and this will be commonplace in the future. And another field that is now very much interesting in the field is the generation of monovalent antibodies that are not modulating, or bispecific antibodies that really are designed to bind to two different things. For example, there's a T-cell immunology or cancer immunology, antibodies that are called BiTEs, that are binding on T-cells on one hand, effector T-cells, and on a tumor cell marker on the other side. And this brings the killer cells to the tumors in order to trigger specific killing. So these are all developments that are now being developed. So maybe the take-home message from that is to try to learn from the knowledge from autoimmunity, how disease is caused, and to transfer that to other disease areas, for example tumor immunology, in order to kill as efficient or damage as efficient as you can. Okay, I'll leave it at that. The people that contributed, list too long to mention everybody.